Disinfectant. Disinfectants are antimicrobial agents designed to inactivate or destroy microorganisms on inert surfaces. Disinfection does not necessarily kill all microorganisms, especially resistant bacterial spores. It is less effective than sterilization, which is an extreme physical and/or chemical process that kills all types of life. Disinfectants are different from other antimicrobial agents such as antibiotics, which destroy microorganisms within the body, and antiseptics, which destroy microorganisms on living tissue. Disinfectants are also different from biocides. The latter are intended to destroy all forms of life, not just microorganisms. Disinfectants work by destroying the cell wall of microbes or interfering with their metabolism. Sanitizers are substances that simultaneously clean and disinfect. Disinfectants kill more germs than sanitizers. Disinfectants are frequently used in hospitals, dental surgeries, kitchens, and bathrooms to kill infectious organisms. Bacterial endospores are most resistant to disinfectants, but some fungi, viruses and bacteria also possess some resistance. In wastewater treatment, a disinfection step with chlorine, ultraviolet, UV, radiation or ozonation can be included as tertiary treatment to remove pathogens from wastewater, for example if it is to be discharged to a river or the sea where their body contact immersion recreations is practiced, Europe, or reused to irrigate golf courses, US. An alternative term used in the sanitation sector for disinfection of waste streams, sewage sludge or fecal sludge is sanitization or sanitization. Properties. A perfect disinfectant would also offer complete and full microbiological sterilization, without harming humans and useful form of life, be inexpensive, and non-corrosive. However, most disinfectants are also, by nature, potentially harmful, even toxic, to humans or animals. Most modern household disinfectants contain denatonium, an exceptionally bitter substance added to discourage ingestion, as a safety measure. Those that are used indoors should never be mixed with other cleaning products as chemical reactions can occur. The choice of disinfectant to be used depends on the particular situation. Some disinfectants have a wide spectrum, kill many different types of microorganisms, while others kill a smaller range of disease-causing organisms but are preferred for other properties. They may be non-corrosive, non-toxic, or inexpensive. There are arguments for creating or maintaining conditions that are not conducive to bacterial survival and multiplication, rather than attempting to kill them with chemicals. Bacteria can increase in number very quickly, which enables them to evolve rapidly. Should some bacteria survive a chemical attack, they give rise to new generations composed completely of bacteria that have resistance to the particular chemical used. Under a sustained chemical attack, the surviving bacteria in successive generations are increasingly resistant to the chemical used, and ultimately the chemical is rendered ineffective. For this reason, some question the wisdom of impregnating cloths, cutting boards and worktops in the home with bactericidal chemicals. Types. Air disinfectants. Air disinfectants are typically chemical substances capable of disinfecting microorganisms suspended in the air. Disinfectants are generally assumed to be limited to use on surfaces, but that is not the case. In 1928, a study found that airborne microorganisms could be killed using mists of dilute bleach. An air disinfectant must be dispersed either as an aerosol or vapor at a sufficient concentration in the air to cause the number of viable infectious microorganisms to be significantly reduced. In the 1940s and early 1950s, further studies showed inactivation of diverse bacteria, influenza virus, and Penicillium chrysogenum, previously Pinotatum, mold fungus using various glycols, principally propylene glycol and triethylene glycol. In principle, these chemical substances are ideal air disinfectants because they have both high lethality to microorganisms and low mammalian toxicity. Although glycols are effective air disinfectants in controlled laboratory environments, it is more difficult to use them effectively in real-world environments because the disinfection of air is sensitive to continuous action. Continuous action in real-world environments with outside air exchanges at door, HVAC, and window interfaces, and in the presence of materials that adsorb and remove glycols from the air, poses engineering challenges that are not critical for surface disinfection. The engineering challenge associated with creating a sufficient concentration of the glycol vapors in the air have not to date been sufficiently addressed. Alcohols. Alcohol and alcohol plus quaternary ammonium cation-based compounds comprise a class of proven surface sanitizers and disinfectants approved by the EPA and the Centers for Disease Control for use as a hospital-grade disinfectant. Alcohols are most effective when combined with distilled water to facilitate diffusion through the cell membrane. 100% alcohol typically denatures only external membrane proteins. A mixture of 70% ethanol or isopropanol diluted in water is effective against a wide spectrum of bacteria, though higher concentrations are often needed to disinfect wet surfaces. Additionally, high concentration mixtures, such as 80% ethanol plus 5% isopropanol, are required to effectively inactivate lipid-enveloped viruses, such as HIV, hepatitis B, and hepatitis C. The efficacy of alcohol is enhanced when in solution with the wetting agent dodecanoic acid, coconut soap. The synergistic effect of 29.4% ethanol with dodecanoic acid is effective against a broad spectrum of bacteria, fungi, and viruses. Further testing is being performed against Clostridium difficile, C. diff, spores with higher concentrations of ethanol and dodecanoic acid, which proved effective with a contact time of 10 minutes. Aldehydes. Aldehydes, such as formaldehyde and glutaraldehyde, have a wide microbiocidal activity and are sporicidal and fungicidal. They are partly inactivated by organic matter and have slight residual activity. Some bacteria have developed resistance to glutaraldehyde, and it has been found that glutaraldehyde can cause asthma and other health hazards, hence orthophthalealdehyde is replacing glutaraldehyde. Oxidizing agents. Oxidizing agents act by oxidizing the cell membrane of microorganisms, which results in a loss of structure and leads to cell lysis and death. A large number of disinfectants operate in this way. Chlorine and oxygen are strong oxidizers, so their compounds figure heavily here. Peroxy and peroxo acids. Peroxycarboxylic acids and inorganic peroxo acids are strong oxidants and extremely effective disinfectants. Phenolics. Phenolics are active ingredients in some household disinfectants. They are also found in some mouthwashes and in disinfectant soap and hand washes. Phenols are toxic to cats and newborn humans. Quaternary ammonium compounds. Quaternary ammonium compounds, quats, such as benzalkonium chloride, are a large group of related compounds. Some concentrated formulations have been shown to be effective low-level disinfectants. Quaternary ammonia at or above 200 parts per million plus alcohol solutions exhibit efficacy against difficult-to-kill non-enveloped viruses such as norovirus, rotavirus, or poliovirus. Newer synergists, low-alcohol formulations are highly effective broad-spectrum disinfectants with quick contact times, 3 to 5 minutes, against bacteria, enveloped viruses, pathogenic fungi, and mycobacteria. Quats are biocides that also kill algae and are used as an additive in large-scale industrial water systems to minimize undesired biological growth. Inorganic compounds. Chlorine. This group comprises aqueous solution of chlorine, hypochlorite, or hypochlorous acid. Occasionally, chlorine-releasing compounds and their salts are included in this group. Frequently, a concentration of less than 1 part per million of available chlorine is sufficient to kill bacteria and viruses, spores and mycobacteria requiring higher concentrations. Chlorine has been used for applications, such as the deactivation of pathogens in drinking water, swimming pool water and wastewater, for the disinfection of household areas and for textile bleaching. Iodine. Acids and bases. Metals. Terpenes. Other. The biguanide polymer polyaminopropyl biguanide is specifically bactericidal at very low concentrations, 10 mg/L. It has a unique method of action. The polymer strands are incorporated into the bacterial cell wall, which disrupts the membrane and reduces its permeability, which has a lethal effect to bacteria. It is also known to bind to bacterial DNA, alter its transcription, and cause lethal DNA damage. It has very low toxicity to higher organisms such as human cells, which have more complex and protective membranes. Common sodium bicarbonate, NaCO3, has antifungal properties, and some antiviral and antibacterial properties, though those are too weak to be effective at a home environment. Lactic acid is a registered disinfectant. Due to its natural and environmental profile, it has gained importance in the market. Non-chemical. Ultraviolet germicidal irradiation is the use of high-intensity shortwave ultraviolet light for disinfecting smooth surfaces such as dental tools, but not porous materials that are opaque to the light such as wood or foam. Ultraviolet light is also used for municipal water treatment. Ultraviolet light fixtures are often present in microbiology labs, and are activated only when there are no occupants in a room, for example, at night. Heat treatment can be used for disinfection and sterilization. The phrase sunlight is the best disinfectant was popularized in 1913 by United States Supreme Court Justice Louis Brandeis and later advocates of government transparency. While sunlight's ultraviolet rays can act as a disinfectant, the Earth's ozone layer blocks the rays' most effective wavelengths. Ultraviolet light-emitting machines, such as those used to disinfect some hospital rooms, make for better disinfectants than sunlight. Measurements of effectiveness. One way to compare disinfectants is to compare how well they do against a known disinfectant and rate them accordingly. Phenol is the standard, and the corresponding rating system is called the phenol coefficient. The disinfectant to be tested is compared with phenol on a standard microbe, usually Salmonella typhi or Staphylococcus aureus. Disinfectants that are more effective than phenol have a coefficient greater than 1. Those that are less effective have a coefficient less than 1. The standard European approach for disinfectant validation consists of a basic suspension test, a quantitative suspension test, with low and high levels of organic material added to act as interfering substances, and a two-part simulated use surface test. A less specific measurement of effectiveness is the United States Environmental Protection Agency, EPA, classification into either high, intermediate or low levels of disinfection. High-level disinfection kills all organisms, except high levels of bacterial spores, and is done with a chemical germicide marketed as a sterilant by the U.S. Food and Drug Administration, FDA. Intermediate-level disinfection kills mycobacteria, most viruses, and bacteria with a chemical germicide registered as a tuberculoside by the Environmental Protection Agency. Low-level disinfection kills some viruses and bacteria with a chemical germicide registered as a hospital disinfectant by the EPA. An alternative assessment is to measure the minimum inhibitory concentrations, mics, of disinfectants against selected and representative microbial species, such as through the use of microbroth dilution testing. However, those methods are obtained at standard inoculum levels without considering the inoculum effect. More informative methods are nowadays in demand to determine the minimum disinfectant dose as a function of the density of the target microbial species. Home disinfectants. The most cost-effective home disinfectant is chlorine bleach, typically a greater than 10% solution of sodium hypochlorite, which is effective against most common pathogens, including disinfectant-resistant organisms such as tuberculosis, Mycobacterium tuberculosis, hepatitis B and C, fungi, and antibiotic-resistant strains of Staphylococcus and Enterococcus. It has disinfectant action against some parasitic organisms. The benefits of chlorine bleach include its inexpensive and fast-acting nature. However, it is harmful to mucous membranes and skin upon contact, has a strong odor, is not effective against Giardia lamblia and Cryptosporidium, and combination with other cleaning products such as ammonia and vinegar can generate noxious gases like chlorine. The best practice is not to add anything to household bleach except water. As with most disinfectants, the area requiring disinfection should be cleaned before the application of the chlorine bleach, as the presence of organic materials may inactivate chlorine bleach. The use of some antimicrobials such as triclosan is controversial because it may lead to antimicrobial resistance. The use of chlorine bleach and alcohol disinfectants does not cause antimicrobial resistance as it denatures the protein of the microbe upon contact.